Hello, welcome back to Bioclass Bites. In this video, we are still talking about the central dogma of molecular biology, but this time we will focus on how DNA codes for proteins. Don't forget to subscribe to my channel and like and share this video. In this video, we will be focusing on the following topics: mRNA transcription, tRNA translation, and protein synthesis. In the previous video, we have established how DNA replication occurs within the nucleus and how it is both semi-conservative and semi-discontinuous. For this video, we will now talk about what happens to that strand of DNA, how it is carried out of the nucleus by RNA, and how it is translated to be made into proteins. Again, I recommend that you watch this video from DNA to protein in 3D. It summarizes everything that we will discuss in this video. Again, I'll provide the link in the description below. Now, for the first part of from DNA to protein processes will be DNA transcription and translation. So this is from McGraw-Hill. Kindly watch this video. This is actually not recommended but required, as it will tell you everything you need to know about transcription and translation. Again, I'll provide the link in the description below. So the first step right after DNA replication is RNA transcription. So this is the process by which DNA is transferred into or copied into RNA. So DNA sequence is copied by a messenger RNA or mRNA to produce a complementary nucleotide transfer RNA strand. So the M here, so this is still an RNA, a single-stranded molecule, but the purpose is to copy, to transcribe, to copy the DNA. So therefore, the letter M is attached at the beginning, making it a messenger RNA. So the goal here is to copy the DNA from the nucleus and then bring it out through the nuclear pore into the cytoplasm. So again, if you still remember, one significant difference between RNA and DNA is that there is no thymine in RNA. It has uracil instead. Okay, so let's review again the difference between DNA and RNA. DNA is double-stranded. Okay? DNA has a missing oxygen here, and DNA has the following nitrogenous bases: thymine, cytosine, guanine, and adenine. It does not have uracil. So the purpose of DNA is to code for the genetic material of the cell. RNA, on the other hand, ribonucleic acid, is single-stranded. It has an oxygen in that missing part, in that missing oxygen in DNA, and it has the following nucleotide bases or nucleobases: cytosine, guanine, adenine. So no thymine, it has uracil instead. DNA is found inside the nucleus. RNA is the one responsible in copying that DNA strand to bring it outside into the cytoplasm where protein synthesis will occur. Now, how does that happen? How does that mRNA transcription happens? I recommend that you watch this video again from McGraw-Hill. It will explain everything you need to know about mRNA transcription. By the way, just to clarify, transcription happens inside the nucleus. Okay? So transcription happens inside the nucleus, then the mRNA copies the DNA because the DNA cannot leave the nucleus. It has to stay there to be protected. But protein synthesis happens outside the nucleus in the cytoplasm. So what will the cell do? It has to copy segments of the DNA using RNA so that the information can be brought outside the nucleus. So that's why we have RNA transcription. Another video that I wish for you to watch will be this one from Amoeba Sisters, wherein they talk about everything from mRNA transcription to tRNA translation up to protein synthesis. Okay, So this is entitled Protein Synthesis Updated. I'll provide the link in the description below. So let's take, for example, this DNA strand for our RNA transcription. So this DNA strand is found in the nucleus, and we want to make a copy of it for RNA so that it can leave the nucleus and go to the ribosomes for protein synthesis. So how will it happen? So mRNA will make a short copy of this DNA strand. Okay, so similar to the DNA replication, complementary bases will be copied. However, the only difference here is that in RNA, they do not have thymine. They do not have letter T. What they have is uracil. So all the letters that are supposed to be letter T here, okay, will be changed to U, to uracil. For example, thymine, the complementary base for that is adenine. Adenine here, it's supposed to be thymine here, but there's no T, there's no thymine in RNA. Now it will be recorded or copied as uracil, as letter U. But then C into G, G into C, C to G, C to G. Then the rest of the strand. Okay, as you can see here, no, thymine is still copied as A, T is into A. So we have another adenine here. It's supposed to be copied as letter T, thymine, but again, no thymine in RNA. So it will be copied as uracil instead. So that goes for the rest of the mRNA strand. So you also have here G will be copied as C, T instead of T will be copied as adenine here. A instead of being copied as T, it will be written as uracil. Then A again, uracil, is into A, C into G. So now you have this short mRNA that can now leave the nucleus through the nuclear pore. Okay, so here they can now leave. So this is the mRNA strand that was copied. Now it can leave the nucleus and go out of it through the nuclear pore. Okay. So that's this part of central dogma from DNA to RNA through transcription. Okay. Now that mRNA that just escaped the nucleus will now be translated, okay, into proteins with the help of tRNA. So the T there, still RNA, but it has a different function now. Okay, so you have here T, which stands for transfer. So transfer RNA. So this uses an mRNA sequence. So tRNA uses mRNA sequence as a template to guide the synthesis of a chain of amino acids. And amino acids are the building blocks of proteins. Okay, so amino acids form proteins. So again, mRNA just came from the nucleus. It went into the cytoplasm wherein it will be translated with the help of tRNA. tRNA will now facilitate the synthesis or the creation of a chain of amino acids which will now become proteins. And that translation occurs in the cytoplasm where ribosomes, the organelles that are responsible for protein synthesis, are located. So these are the three steps that are involved in RNA translation. So initiation or the start usually is based on the code AUG, GUG, or UUG. So these are the start codons. This will trigger or this will command that what do you call this? That the start of the amino acid chain should begin. Then elongation, so those are all the amino acids that will be brought into the chain corresponding to their codon. Okay, so later we'll talk about that. And then once our amino acids have elongated, then we will have termination. So that's the reading of the final mRNA codon that will tell the ribosome to stop creating or synthesizing the amino acid chain or the peptide chain. So initiation start, elongation, it elongates the amino acid chain. Termination, it stops the process. Okay, so the stop codons are UAA, UAG, and UGA. So codons, so this is how the, if you still remember, um, if you still remember, we've talked about this, that each of these nucleotide base will become important in creating the amino acid. Now, in order for them to be utilized, they have to be read by threes. Okay? So the genetic code is actually based on how these three codons are, these three nucleotide bases are read as one codon. So the code defines a mapping between a trinucleotide sequence called codons that are involved in creating amino acid. Each triplet of nucleotides specifies a single amino acid. So these three letters will become one codon that is actually corresponds to one amino acid. So they are read by three. They are always read by threes or grouped by threes. So we call that codon. So let's look at this mRNA strand as an example for RNA translation. So what's going to happen is that they will be read by threes, again, no, as codons, and they will correspond to a specific amino acid which will form the protein. Okay? So how do we do that? So a tRNA anticodon will be attached to these mRNA codes, and then they will bring the appropriate amino acid in the amino acid chain. So when you see the codon AUG, that is actually your start codon, if you still remember. So AUG, when found at the beginning, actually activates the start of the amino acid chain. So if you still remember, no, you have your start codons, AUG. So this is the most common start codon, mostly used by eukaryotes. While some prokaryotes, they use GUG or UUG, but most common is AUG. So it means that you start the amino acid chain, followed by CGG, which corresponds to the amino acid arginine, AAU, which corresponds to the amino acid asparaginine, GCA for alanine, and then if you find AUG um, in the middle of the mRNA strand, it will not you will not write here start, but you will write here methionine instead. Okay? Then CAU for histidine, and then UAG, that's a stop codon. So again, you have start and stop codons. So your stop codons are UAA, UAG, and UGA. So it means that the amino acid chain is done. It will now leave the ribosomes. It will now become a protein. Now, your proteins are very important because they have a lot of functions in the body. They could act as enzymes, okay, for biochemical reactions. For example, the enzyme the protein pepsin that's found in your stomach is actually a digestive enzyme. You have antibodies that are made up of proteins such as immunoglobulin that fight off infection. You have proteins that are part of the structures of some cells. You also have proteins that can act as hormones. For example, insulin is a hormone that's important for the regulation of your blood sugar level. And even transport proteins that are found, that are important in cell-to-cell transport or the transport of materials in and out of the cell. So this one is your protein now, then it can now leave the ribosomes. Now here is a guide to 20 common amino acids. So note that there are over 500 amino acids found in nature, but only 20 of those are directly coded by our human genes. So examples of those will be alanine, glycine, isoleucine, leucine, proline, and then sometimes it's recommended that we just use the first three words, the first three letters of their names because it's quite long. So as you can see here, we only use the first three letters of their names, okay? Now, you are not, and then below that are the DNA codons, okay, that codes for them. But do not worry, you are not required to memorize all of those. That's quite difficult. You will be presented instead with this mRNA codon amino acid chart or variations of it when you take assessments. So one thing is that you have to learn how to read this chart. So how do you do that? So for example, AUG, AUG is read this way. The first letter or the first base is found on the left side, A. Then the second base is found here on top, U. And then the third base will be found here on the right side. So A, U, G. Okay, that's your start codon. Okay, start codon. So that's what you're going to write. Instead of methionine or met, you will write start because that's the beginning of your genetic of your amino acid chain. Okay, but if you found this at the middle of the strand, then you write methionine. Another example, GCA. So the first letter is here, G. Then C is the second letter here, and then A. Oh, it's being covered. GCA corresponds to alanine. So you write alanine or ALA as the amino acid. How about UAG? U A G. So it is a stop codon. So you will see here in the amino acid chart, you have the following stop codons, UAA, UAG, and then UGA. So when you see here, when you see it, it means that stop. You stop the amino acid chain, it's done, the protein is finished. But again, no, if you see AUG at the beginning, what you write is start. Okay, so that's the start codon. So you write start, but if you find here, if you find it in the middle, just like this, you write here start, but if you find it at the middle, you write methionine. Okay, so that's how you use the amino acid chart. To learn more about the roles of the amino acids and proteins that are found in the body, I recommend that you visit this from Science Learning Hub. I'll provide the link in the description below. So let's summarize again the steps of the central dogma. First would be, of course, DNA replication. We've talked about that, okay? Wherein DNA replication is both semi-conservative and semi-discontinuous. And then the goal there is to make copies of the DNA strand. Then it will be followed by mRNA transcription. So wherein a short segment of the DNA will be copied by the mRNA because the DNA cannot leave the nucleus. Only the mRNA can leave the nucleus. So once the code has been copied, mRNA will now go out of the nucleus and go to a ribosome. Okay, wherein that mRNA or that complementary strand of the DNA will be read by these. We call them codons. Okay, we call them codons. Okay, and they correspond to a specific amino acid. So, again, also methionine, but if it is found at the beginning, we write their start. And then you have threonine, and the rest glutamine, and cysteine, and then the rest of the amino acid chain until you reach a stop codon. Okay, which tells the ribosomes to stop the creation of the amino acid chain. So this is the summary of everything that we have tackled so far. Now, if that particular DNA strand, okay, that will eventually be created into an amino acid chain, if that particular DNA strand is heritable, it means that it's passed on from parent to offspring, and they happen to code for a specific trait, we call that segment of DNA a gene. Okay? So to learn more about how strands of DNA or how segments of DNA are considered genes, I recommend that you watch this video from Stated Clearly entitled What is a Gene? This is very important, and I'll provide the link in the description below. Then this video, which summarizes everything that we've discussed, this is from Khan Academy, DNA replication, transcription, and translation. Kindly watch this video, and I'll provide the link in the description below. So that those are the important parts of the central dogma of molecular biology. It starts with with DNA replication within the nucleus, transcribed by the RNA to be carried out into the cytoplasm for translation, eventually to be made into amino acids that eventually codes for protein. So now what's protein and what's protein synthesis? So that's the process by which cells build proteins. So again, proteins are large compounds made up of amino acids. So those are your valine, alanine, methionine, all of those. And then if you put them together, they can be arranged in a linear chair and joined by peptide bonds. So these are representations of those amino acids. So one amino acid plus another amino acid, if they are bonded together, they become a dipeptide. Now, several dipeptides can become a di means two, can become a polypeptide chain. Poly means many, so many chained amino acid, which will eventually become your protein. So again, I've mentioned this a while ago. So what are again the functions of proteins? So they act as enzymes that actually catalyze biochemical reactions and that are responsible or important for your metabolic processes. They also have structural and mechanical function such as actin and myosin, which are found in your muscles, and proteins found in the cytoskeleton of your cells. For cell signaling or immunoglobulins for cells to recognize each other, immune response, cell adhesion, and even the cell cycle. And animals' diets, since animals cannot synthesize all the amino acid, so therefore we need to obtain it from the food that we eat. So it's important that we have a good source of proteins in our diet. Now, here are recommended videos for you to watch. This is from TED Ed, okay, a TED talk conducted by James Watson, one of the the ones who proposed that double helix model of the DNA. So this is entitled How We Discover DNA. I'll provide the link in the description below. This one is another video wherein the two of them, Watson and Crick, explained how they discovered the DNA. So I'll provide the DNA double helix model. I'll provide the link in the description below. And this one is still from TED Ed. Rosalind Franklin, the DNA's unsung hero. So kindly watch this video. It's required so that you will see where the inspiration of Watson and Crick came from and why Rosalind Franklin is slowly being recognized as also another one who discovered the DNA double helix structure. So watch this video and I'll provide the link in the description below. That ends our video. I hope you learned something new today. Don't forget to subscribe to my channel and like and share this video. Till next time, goodbye.